I'm Joseph Silva. Hi, I'm Joseph Silva. I come from the University of California, Merced, the Department of Material Science and Engineering. My group works on solar selective coating, and solar selective coating helps reduce the cost and improves the efficiency of current solar devices. So in particular, we are working with low temperature solar selective material such as aluminum. Aluminum is cheap and it's really easy to process. And what we're trying to prove is that if you introduce an oxide layer to aluminum and get a nice linear rate of diffusion between aluminum and oxygen, that you can have great light penetration, and it can actually act as a great light absorber and heater. So currently, if you've seen those black thermal panels on just people's houses, that can get you up to about 35 degrees Celsius. That's okay for residentials, but if you want industrial grade heating, you would want about 100 to 200 degrees Celsius. And aluminum is great because it can get really hot without melting, and it gives off a good amount of heat. So once the light passes the oxide layer, you'll get a bulk of aluminum, and the reflectivity properties of aluminum will keep light trapped within the material and heat your thermal, will heat your, either whether it be water, whether it be oil. Water at that temperature would turn to steam, so a lot of companies like steam power, or you can have thermal, or oil, which can transfer the energy throughout the, wherever you want it to go, you can just pipe it through. So currently we are using fixed second law as a method for any given X to determine concentration at different depths. We have the diffusion of oxygen across the grain boundaries, and we have this diffusion of aluminum across the grain boundaries. And as you can see here, aluminum diffuses a lot faster than oxygen. So we're trying to introduce different methods to slow down that aluminum diffusion rate, so it doesn't just push the oxygen out of the aluminum oxide layer. One way we're doing that is while we're heating aluminum, we introduce sodium bicarbonate, which once it's heated up at the diffusion rate, or the diffusion temperature of aluminum, it gives off CO2. CO2 will help slow down that self-diffusion of aluminum. And this is a graph of a high temperature material, but it's close to what we want for this low temperature material. We don't have a really great method of modeling for our material right now. That's why we're trying to use NanoHub to actually, and NanoHub, we're trying to use the Python as the language we're using in NanoHub, and we want to try and use this equation to give us that nice graph of oxygen penetration through the aluminum. Currently, right now, we're just using SEM, scattering of light microscopy, electron microscopy, to get the concentrations at different depths. Different depths correspond to the different kilovolts. This is 5 kilovolts. You can see that the aluminum to oxygen is pretty close. At 10 kilovolts, the aluminum to oxygen you're getting a little more aluminum. And at 15 kilovolts, you're getting a lot more aluminum, which is pretty much what we want, because we want to keep the light in as it passes that oxide layer. And yeah, our next step is just to introduce more, some other dopants, which helps the diffusion rates of aluminum and oxygen.
Hi, I'm Rosalind Xu. I'm from the Department of Cell and Developmental Biology here at UIUC. The poster I'm presenting today is about the study of dynamics of ORC1 and ORCA at telomeres and their role in DNA damage response. Basically, both ORC1 and ORCA, there are known proteins that involved in DNA replication initiation. But what we're trying to address here is their additional role in the cell especially at telomere. From this image, this is a previous publication from a lab. We can see this is an immunofluorescence image. We can see that the signal of ORCA, ORC1, and telomere are actually co-localized, which indicated by these white spots. So we can see that these proteins localize at telomere, and basically protein does not go to a place that does not have a function. Besides that, we also found that the localization of these two proteins with telomere is specific in cells that use a different mechanism for telomere lengthening, which is called alternative lengthening of telomeres, which basically utilize homologous recombination. Besides these evidence, there are other reference which indicates that both ORC1 and ORCA, they have a consensus phosphorylation site which indicates that they might participate in the DNA damage response mediated by ATM and ATR dis2 kinase. So based on this evidence, the aim of this study is, the first is to study the role of ORC1 and ORCA at telomeres, and whether they have a role in DNA damage response, and how they interact with DNA, I would say more specifically with telomere. And then the bottoms are several approaches that we wish to address our questions, such as right now we're using truncated mutants to see which is the minimum domain required for these two proteins to associate with telomere. And we also want to see whether these two proteins directly interact with the telomere DNA sequence, or whether they interact with the telomere-associated protein, which is the sheltering complex instead. So this is my study.
Hi, my name is Amanda Pentecost. I am a second-year PhD student at Drexel University. I'm working in two different labs with Dr. Kara Spiller in biomedical engineering and Dr. Yuri Gagotsi of the Nanomaterials Institute at Drexel University. So I'm interested in combining the fields of materials engineering and nanomaterials and applying it to biomedical applications. So I'm working with wound healing and macrophages. Macrophages can be divided into two different categories: classically activated or pro-inflammatory, like the M1, or alternatively activated, including M2A and M2C, which have a pro-healing response. So in things like chronic wounds, such as diabetic wounds or venous ulcers, you have wounds that do not heal. And they're stuck, this is because they're stuck in the M1 phase. They're stuck in inflammation. You get a cut and it just does not heal. So what I would like to do is take these people who have this cut and use a therapy targeting macrophages to get them to go into the pro-healing state. And this would be, even if it's scar formation, but something that closes the cut and stops the inflammation. So I propose to use a nanomaterial called nanodiamond. Nanodiamond is interesting because it has the same properties as bulk diamond, but at a nanoscale. It has the same chemical structure and it's very hard. What's also interesting about it is that it has a 5 nanometer average particle size, and it's proven to be very biocompatible, and also has very rich surface chemistry, which is strange because we usually think of diamond as being very inert. So it actually, as you can see here, has lots of carboxylic acid groups and hydroxyl groups on the surface. And we can do a lot of surface treatments, such as purification, to increase the number of carboxylic acid groups, or we can do amination. So over here I have pictures of two types of modified diamond. This one is modified with octadecylamine. So it has an amine group on the surface, and what was really strange about this is that it's blue and hydrophobic. So neither the diamond nor the octadecylamine have these properties in itself. But when you put them together and you covalently bind them, you get this blue hydrophobic particle. So another thing that we can do is physiosorb rhodamine on the surface. And this rhodamine is called tamarine, which is only found by van der Waals bonds. So the title of my project here is a little bit misleading, as I haven't gotten to do looking at using nanoparticles and macrophages. But what I do is investigate a drug called dexamethasone and its effect on macrophages. So for simplicity's sake, since this is my personal start to the field of biology, is we took THP1 cells, which are type of macrophage line or monocytic cell line derived from patients with leukemia. And we differentiated it first into an undifferentiated macrophage, and then into these 3 different phenotypes, M1, M2A, and M2C, using procedure found in the literature. So these were seeded. We added different doses of dexamethasone, a high dose of 10 to the negative 6 molar, and a low dose of 10 to the negative 8 molar of dexamethasone. And what we think this drug will do is a steroid. And as you know, steroids are known to decrease inflammation. And so we wanted to see if this was really what was happening and if it was actually inducing a pro-healing response in addition to decreasing inflammation. So we used RT-PCR to quantify this using known gene markers for the 3 different major types of macrophage phenotypes. And this is my first shot, so don't laugh too much at my scatter. Oh, sorry. But we did find that there is large downregulation in 2 of the M1 or pro-inflammatory markers. And actually at a low dose, you can see this M1 here actually switches, has an upregulation of CD163 and actually switches to the M2C phenotype. So this is an increase in the regulatory behavior. Basically, in the future, I would like to put this drug onto the diamond surface and look at a couple of different things. I want to look at how the drug is taken up by the macrophage. Is it receptor-mediated endocytosis, which is how most cells take up the drug, or is it phagocytosis? You can think of macrophages like Pac-Man. They just go in and they eat everything. Which is why we want to use nanoparticles to target these macrophages. So I'm also going to look at the adsorption and desorption properties of dexamethasone onto diamonds, and characterize the phenotype using different types of diamonds. And of course, I would personally like to review all of this with human macrophages so that it becomes biologically relevant.
All right, so I'm Claire McGee. I am from the University of Illinois, and I work in Professor Yi Lu's lab. So basically what I'm going to be talking about today is using a glucose meter to monitor something else like lithium in this case. Now the reason I wanted to monitor something like lithium is because lithium is used to treat bipolar disorder for about 23% of patients, and it's used specifically to depress the manic state. So one of the problems with using lithium is that it has a very small therapeutic window, so it's about 0.5 millimolar to 1.2 millimolar in serum. Above that, we see toxic effects, and below that, it's not effective. And so we want to monitor this drug, and also lithium's role in reducing the manic state is not completely known, so if we could do cellular studies, that would also help us to further understand the disease and potentially create new drugs for afterwards. So in order to do this, in order to turn something like a glucose meter into something that can sense lithium, we're going to use something like a magnetic bead. We start off with a magnetic bead that is attached to DNA, which will react specifically with a target. And so this DNA can be either an aptamer or it can be a denazyme, which is a catalytic strand of DNA. And then lastly, that DNA strand will be attached to a protein called invertase. Now, this DNA strand, when introduced to the target, in my case it would be lithium, would cause a release of your invertase protein, and that invertase protein can then change sucrose into glucose. Now this glucose meter will read for glucose, but it will not read sucrose. So in that way we can get a direct correlation between the glucose sensor reading and the amount of target that we initially have. And one of the reasons that we want them on magnetic beads is because we want to be able to remove anything that is non-reactive, or the invertase that is still on magnetic beads, from the solution, so that we get something that is directly proportional to the target in glucose reading. All right, so how are we going to do this, and what is the strand of DNA that's going to be the interlinking basically target reader? And that's going to be a denazyme. So a denazyme is, again, DNA that is catalytic. We program it to have is this RNA strand right here, because RNA undergoes hydrolysis more easily. So these nucleotides are all programmed in, but then we have this region right here that is random. which means that we basically send it into a company. They add all the nucleotides together, and they will randomly synthesize. So if you have something like 25 random nucleotides, then it gives you 4 to the 25th different types of sequences. So that means each one of these sequences is going to fold a little bit differently, or it's going to interact differently. And so of these, some of them are going to react with lithium in one way, some of them are going to react in another, some of them will react with calcium, sodium. There's just a plethora of things that they could do. So what we do then is, if we have this DNA, then we can come back over here and we can see that once it causes a hydrolysis reaction, that will cause the break of the strands, and that will cause dehybridization over where we have a low melting point, and the two strands can separate. That would mean the release of a fluorophore if we were going to do a fluorophore quencher model, or it could cause the release of invertase, as we would see up here. So in order to get something or find out what this random region is right in here, then we can go ahead and use a technique called in vitro selection. So in vitro selection, again, we start off with something like 10 to the 13 different types of sequences, so a very high number, and we expose them all to some sort of negative selection or some sort of conditions that contain ions that we don't want our DNA to be reactive with. What that does is it causes, again, the cleavage reaction. So we have two different sizes of DNA that are floating around in solution. Again, if we take around 20 nucleotides off of something that weighs about, or something that is about 110 bases, then we're going to get a significantly shorter strand, so we can just separate this via gel electrophoresis. So we separate then the things that are reactive with everything we don't want from the things that are not. And then what we do is we take what is left unreactive and we react it with our target. And then let's say that this now reacts with lithium, which is what we want. We then keep all of these sequences that are reactive, and we discard everything we don't using, again, gel electrophoresis. And so then we can take everything that was unreactive and use PCR amplification to then go back into this round up here with a different set of DNA or a more limited set of DNA and continue throughout these rounds. So the progress that I've made on this so far is by going through about 20 rounds of selection. I have two different pools, one that has around 25 nucleotides in here and one that has about 50 nucleotides in the random region. So what we see is a general increase in the amount of DNA that is cleaving in the presence of lithium to about 10% in the later rounds. So that means our sequences, or our general pool, has sequences that are responsive to lithium, which is ideally what we want. Now, if we go ahead and we look at this and we compare it to other monovalent species like sodium, potassium, cesium, and rubidium at the same concentration of lithium, we can see that this is clearly more reactive with lithium than it is reactive with other monovalent species, which means we're heading towards our goal. Where we can take this then is to go ahead and clone these and sequence them and find out what exactly is in this region that is making it reactive specifically with lithium. All right, thank you.
So my name is Dylan Dogwis, and I'm presenting a project that uses the graphite from pencils and through a variety of treatments functionalizes it into a supercapacitor. So here we see a couple of the different treatments that we've gone and performed thus far. First off, we have our annealing treatment, which we found that if we take the graphite from a pencil, put it into a furnace with flowing argon, and anneal that sample at 1100 degrees Celsius for 5 hours, we see a 130,000% increase in the capacitance of that material. From there, we also studied the effect of composition of the actual graphite itself on the capacitance of the material. So pencil graphite is actually a little bit misleading because it's not pure graphite. It is a mixture of graphite and clay. So your standard number 2 pencil has a certain ratio of clay to graphite so that it can appear on the standardized tests. So what we did is we went and got drawing pencils that have a higher graphite and they're softer. And we also went and got higher clay ratios, which are a little bit harder and a little bit lighter. Excuse me. And what we saw here was kind of predictably was a large increase in energy storage in the high graphite samples. But curiously enough, we also saw very well-defined peaks of these curves in the higher clay samples. It's kind of hard to see in this figure because it's put on the scale of the high graphite, but these show a little bit more defined edges as to the redox peaks in the CV curve. And a quick explanation of CV curves really quickly is it stands for cyclic voltammetry. And what we're doing here is we sweep from 0 to 1 volts. And we do that because if you go higher than 1 volt, you'll split water and get noise in your data. But we sweep from this electric potential and we measure the current that's coming out of the solution of these 2 electrodes. And so we have one electrode that's platinum, that's our working electrode that's supplying the potential. And then the counter electrode, which is the thing that we're measuring. And so when we go along this potential and we see zero current, that means there's no electron transfer going on between the solution and the working electrode, the thing we're measuring. Once we start to see it increase at around 0.5 volts, is what we've hit an electric potential, which is allowing for electron transfer and electron current. So that's just the flow of electrons. And so we're seeing that current, and so we're seeing a charge increase as we go up, and then this is the oxidation that happens, and then we sweep back from 1 to 0 and get reduction. So basically, we're seeing that there's a direct correlation between the energy storage of the material and the area of this curve. Now going back to the original treatments, one other treatment that we did was we grafted carbon nanofibers onto the surface of this pencil, or the pencil graphite. This increased the surface area and increased the capacitance slightly, but what we were more interested in was the durability of the material. So we saw with the carbon nanofibers that the durability of the material after repeated cycles, the capacity dropped off less than when it was bare and just by itself normal pencil graphite. The last treatment that we explored was grafting conductive polymer onto the surface of the pencil graphite. And what we did there was graft conductive polypyrrole onto the surface, and we saw a 300% increase in capacitance. And that is from the original annealing capacitance, so we improved this great improvement even further. So from this study, we've actually explored a couple of treatments of this carbon material to improve its energy storage for potential devices such as supercapacitors or perhaps fuel cells. And in future work, we wish to do some XPS testing as to what is really going on at an elemental level and what is causing these increases in energy storage. Thank you.
Okay, so hi, my name is Kenry. I'm from National University of Singapore. I'm currently a second-year PhD student. So the project that I'm currently working on is titled Viscoelastic Effects of Silicon Gels for Cellular Traction Force Measurement at the Micro and Nano Scale. So basically, the objective of this project is to characterize the viscoelasticity of silicon gels in order to evaluate the suitability for traction force measurement. So normally when people want to measure traction forces, people tend to use elastic materials with minimal viscous response. However, in recent years, there has been increased use of silicon gels, which is a viscoelastic materials for this purpose. And then most of the characterization work done on silicon gels are performed on bulk materials. However, for most of the cell traction force assays, only the substrate surface is deformed when cells exert nanonewton forces over the underlying substrate for a micrometer length scale. So there arises a need to characterize the viscoelasticity of these silicon gels at the micro and nano scale. So what we do is that we actually evaluate 5 different types of gels based on the different stiffnesses. And then we use atomic force microscope to probe the stiffness of these materials in order to evaluate whether it exhibit these time-dependent and strain rate-dependent responses or not. So based on the available theoretical models like Hertz model, DMT model, we actually extract the elastic modulus of these gels from the force indentation curve. And these are some of the results displayed here. After evaluating the elastic modulus of these gels or the stiffness of these gels, basically we move on to characterize more about the viscoelastic behavior of these gels. So using the same instrument, atomic force microscope, we basically characterize the creep response of these 5 different types of silicon gels. And based on this Voigt SLS model, we actually extracted all the viscoelastic parameters of these silicon gels. And lastly, we seeded fibroblast cells on these 5 different types of silicon gels and evaluate whether this traction force exerted by the adherent cells will induce permanent or non-elastic deformation or not on these silicon gels. And eventually we concluded that silicon gels with high stiffness and elasticity, they basically exhibit short characteristic retardation time, and they will be more suited for cell traction force measurements. Thank you.
So my name is Murali Yellappu. I'm from University of Tennessee Health Science Center, and I'm a student faculty over there. So today my poster, it deals with more of magnetic nanoparticle formulation and human serum protein interactions. So what we did, this project we started 5 years ago to improve the drug delivery and imaging capability of magnetic nanoparticle formulation. So what we did, we generated unique double-layered formulation that can be used for both drug delivery and imaging with antibody-guided therapy. So here the poster, it gives more information what happens if the nanoparticles are incubated with human serum, because that determines whether the particles can be reached to the tumor or intended target. So what we did see in this work, so when we incubated nanoparticles with the human serum, the particles are highly bound to the protein called APOE1, so which indicates that the particle formulation has more bioavailability or more in the circulation that can be reached to the tumor for therapy application. And moreover, this particle formulation size, it's not altered, and also the particles, it did not induce any hemotoxicity to the human red blood cells. Additionally, the particles, when incubating with the human serum protein, they did not alter inherent properties such as MRI properties and hyperthermia and other applications, so because it has higher magnetization saturation values. So overall, this poster, it speaks like this formulation is it has more stable in the human serum, and that can be used for human therapy applications in the near future.
My name is Zainab, and I am with the Department of Biochemistry at University of Illinois at Urbana-Champaign, and my PIs are Dr. Tegjib Ha and Dr. Deborah Legban. My project basically is trying to address the clear gap of knowledge between the how the single molecular forces act or influence the cell receptors, which indirectly or directly influence the cellular function. To address this, recently HiLab developed novel DNA nanostructures, and we call them TGT, or tension gauge tether. How these work is that if the tension tolerance of this TGT is less than the force applied by the transmembrane receptor, the TGT ruptures, as it can be seen here, and which directly or indirectly influences receptor-regulated cell functions. And a readout for that in our case is the adhesion of cells on this TGT modified surface. However, if the tension tolerance of these TGTs is greater than the force applied by the transmembrane receptor, the TGTs remain intact and the receptor-regulated cellular functions are activated. Another major key player in my thesis, in my project, are cadherins. Cadherins are transmembrane proteins which are involved in cell-cell, which are involved basically in cell-cell junctions. And if there is an aberrancy of cadherin expression, then we can witness something as majorly wrong as cancer metastasis. So how do I combine these two and try to address the problem is that we have engineered cadherin TGT in our lab, and we are using the system to measure the tension tolerance of cadherin bond. The design of cadherin TGT is very simple. We have a 22 base pair DNA double strand which is conjugated to a protein G, which links to any sort of Fc domains to create of E-cadherins or any other proteins for that matter. And then we have a 6 nanometer PEG linker, which prevents the steric hindrance of the TGT from attaching it to the surface, and this has been anchored to the surface using neutravit and biotin. The surface is passivated by PEG. So our preliminary results show that this design actually works and that cells are able to adhere on the TGT modified surfaces. These are the DLD1 cells. However, we're not able to see MDCK cells and MCF7 cells, which are important in cadherin studies to these cadherin TGT modified surfaces. So we have tried to address this issue, and what we have been able to conclude is that the E-cadherin density on the control surfaces without the TGT is much higher than the E-cadherin density on the cadherin TGT surface. So currently we're working on increasing the cadherin density on the E-TGT surface. My second aim is to determine the critical tension required for E-cadherin mediated mechanotransduction. For that we will be using magnetic twisting cytometry. In this experiment, we cover our cells with these magnetic beads that are basically coated with the ligand of our choice, and then we apply a torque on it, and if there is a stiffening response, that is the amplitude of the bead movement decreases, it is a readout for mechanotransduction, as it can be seen here. However, if there is no stiffening response, we see no mechanotransduction has happened. So if for an example of that behavior was seen when we applied the magnetic bead and the cells were coated with E-cadherin, then we saw a stiffening response. That was a dampening in the amplitude, and that was correlated to the increase in the stiffness of the cell. However, when the cells were coated with PLL, polyl lysine, which is a non-specific molecule protein and is used as a control, we did not witness any dampening in the amplitude of the bead displacement, which meant that no mechanotransduction has happened. So we will use basically the stiffening of the bead or the reduction in the amplitude as a readout of our mechanotransduction. We will be coating our beads with cadherin TGT of different tension tolerance, and we will see where we get the stiffening response, and we will use that to determine the critical tension required for cadherin mediated mechanotransduction. The other part of our design is that we we'll be using the system to study the crosstalk between integrin and cadherin and the response of the global mechanotransduction. Our beads would be coated with cadherins, and our cells would be coated on surfaces which are functionalized with integrin TGT, and then we'll see how does the mechanotransduction basically change the activation of the integrin TGT, and that's the description of my project. Some of it is ongoing and some of it has to be done.